So chapter 12 in the physical or digital textbook just gives you a bit more information about sensory receptors and sensation, which are really part of what your nervous system does. But now we're just going to go into a bit more detail on some of those specific receptors. So this lesson will be about sensory receptors in general and specifically about photoreceptors. So the first thing we need is a bit of vocabulary. So the first descriptor up there is about what a sensory receptor is. Sensory receptors are sometimes terminals, so the end of a nerve, or cells that detect sensory information. You have a lot of different types of sensory receptors. What they all have in common is that they detect this information. They are attached to a sensory neuron, and that stimulation will cause an action potential like we saw in the last section. Now, just as a reminder, because it's really important, a stronger stimulus, so stronger stimuli, which is the plural of stimulus, cause more frequent action potentials. They don't make the action potentials bigger, they make them more frequent. When we watched the video, the guy talked about it being a buzz, and that the buzz didn't get louder, it just got faster. It went from bzz, bzz to bzz, bzz, if the stimulus was really strong. So you have all these types of sensory receptors, and I'll list some of them as we go through this lesson. But they all have this function in common. They're detecting sensory information, they're attached to sensory neurons, and we know where sensory neurons go. They come from the receptor and go towards the central nervous system. If it's a reflex arc, then it's a simple pathway right through the spinal cord. If it's a more complex thing, information goes up to the brain. Now, sensation is the word that we use to describe nerve impulses reaching the cerebral cortex. Your brain gets the information. Perception, though, is your brain's interpretation of what that sensation meant. And the two things are not the same. Your body can sense something, but then you can perceive it a different way. The first question on your sheet asks about eyewitness reports at a crime scene. And if you ever watched detective or police shows, you'll know this is a continual source of frustration, where everybody was there for the same thing. It wasn't like you could have seen something different because the same thing happened for everybody. So it's not like the light was different and the stimulus that went in your eye was different, or it's not like the sound was different and what went in your ear was different. But the perception of what happened, your interpretation of what happened, might be different. So sensation is about nerve impulses actually getting there. Did this sensory receptor pick up information? Did it send that information along through a sensory neuron? Perception, though, is how do we interpret that? Now, your nervous system does something cool for you called sensory adaptation. Your brain is continually trying to make your life easier by skipping over things or by filling in missing pieces. There are so many cool brain tricks, optical illusions that are based on this principle, that your brain tries to fill in information that's missing, tries to guess ahead as to what's coming next, or tries to take a shortcut to make it faster for you to process information. Sensory adaptation is one of those. It's where your nervous system will try to filter or eliminate redundant or insignificant information. So for example, there is a clock on the wall. Everyone be quiet and listen for the tick. Okay, there, I can finally hear it now. It took me a second and I had to look at the second hand moving, and if you're far away, it might even be so quiet that you can't hear, but maybe those of you who are right by the wall could hear the faint ticking of the clock. But we had to stop. I had to stop, and I had to be quiet, and I had to focus on the thing that I knew was making a sound, because my brain was ignoring the sound. It was this ambient noise that's always there that was really not important to what was going on. Now, if a bomb was about to explode and I only had seconds left, maybe the ticking of the clock would be actually really super duper important, and I would focus on it. There's another really good one that's happening right now to all of you. Stop for a minute and ask yourself, what do my clothes feel like touching my skin? I hope there are clothes touching your skin, but I'm gonna bet that you aren't sitting there thinking, okay, my sock is touching my foot, my pants are touching my leg, my shirt is touching my torso. I don't think that that's a conscious awareness thing that you have until you stop and think, can I feel my clothes? Oh yeah, there they are. But it's not information that you generally need as the most important thing in the forefront of your thoughts. So, when you have constant stimuli, so something that's always happening, that's repetitive, like the ticking of the clock or the feeling of your clothes touching your skin, it can lead to a bit of decreased sensitivity in the receptors and the neurons that are dealing with that. And so in the long run, probably less ACH, which is acetylcholine, or whatever neurotransmitter is involved in the pathway in the synapse. Your nervous system is trying to accommodate what's going on. It's trying to filter out the stuff that you don't need so you can focus on what is the most important thing. Now, if anyone has questions about any of these terms, you want to let me know? Okay. So like I said, there are different types of sensory receptors. And we're going to do an overview of a few of them and go into a bit more detail about some other ones. The first category of sensory receptors that we'll look at are chemoreceptors. Like their name suggests, chemoreceptors, they're stimulated by some kind of chemical substance. And so I'm indicating which type of stimulus each of them need, because that's really the thing that differentiates the different sensory receptors. When a chemical substance binds to one of these receptors, it causes depolarization, initiates an action potential. That's what happens in all sensory receptors attached to sensory neurons. So a few examples of chemoreceptors that you have. First could be taste. So over here on the left, there's a picture of a bunch of different tongues and where you may or may not be able to detect different flavors. But you have taste buds on your tongue. Their job is to detect different food particles that get dissolved in your saliva. And when those food particles bind to the taste bud, it's attached to a neuron. So this picture over here tries to illustrate how you've got these little pores on the surface of your tongue. That's where the food particles can go. They're attached to these taste receptor cells, and then there is a nerve that will go to your brain to tell you that was chocolate, or that was lemon, or that was gross. Don't put that in your mouth. But that's how a chemoreceptor works. Chemical substance binds to the receptor, initiates an action potential in the nerve that it's attached to. Smell is another sense that uses chemoreceptors. So the sense of smell is called olfaction, which is why they're called olfactory receptors. You have these in your nose, like this person is showing here. Chemical molecules get suspended in the air, and so when one of those molecules attaches to the olfactory receptor in your nose, same thing will happen. Will initiate an action potential in the nerve that's attached to that sensory receptor. You also have some internal senses that use chemoreceptors. So for example, you have things called osmotic receptors. Maybe you remember them from Bio30E, dealing with water. But they'll detect levels of water and other substances in the blood so that your brain knows whether you need more water and you're thirsty and you need to retain water, or whether you can excrete water, let it go. There are also receptors in some of your arteries, specifically in the aorta and the carotid artery, that will detect blood pH. And so they'll work based off of hydrogen ion concentration. So these are some examples of chemoreceptors. They all work the same way. Chemical substance binds to the receptor. Receptors attached to a sensory neuron that brings information to the nervous system about what the chemical molecule was. Now you also have a category called mechanoreceptors, and there are several examples in this category. There's a picture off on the side that's representing your skin. You do not have to be able to name all of these. I just wanted to give you a visual of how many different types of receptors you have in your skin. On the picture, the top is the surface of the skin, and then there's all this stuff going on underneath the surface of the skin. You have receptors in your skin for things like touch, for things like pressure, for things like pain, and so a bunch of different types of receptors detect different types of touch stimulus on your skin. You'll see up here there's free nerve endings that are for pain, heat, and cold. You have 3 over here, Merkel disc, Krauss end bulbs, and root hair plexus that are all for touch, but different degrees or types of touch. Then over here, you've got these Pacini corpuscles and Ruffini endings that are for pressure. So pushing on the skin, and it depends how deep and how hard things are pushing as to which ones are stimulated. So you have mechanoreceptors. They're detecting mechanical pressure, so actual physical touch on your skin. Whereas our chemoreceptors required a chemical substance to bind to them. These mechanoreceptors work based on some sort of movement. Something has to move for them to be stimulated. Now, hearing falls under the category of mechanoreceptors. We're going to talk about hearing really specifically, but essentially, you have these cells inside of your ear that detect sound waves, because sound waves make things move in your ear. So again, it's based on this movement, this mechanical change that's happening. We'll also talk about balance. Balance and hearing both have to do with your ears, but with different cells in your ears. This time, it's movement of liquids that we're looking at, and also movement of little tiny rocks. So we've got liquids and rocks moving inside your ear that tell you that you're hearing things and that your head is moving position and that maybe you're out of balance. You also have mechanoreceptors for body position. So you have receptors in your muscles and tendons and joints that will detect movement, specifically stretching, contraction of those types of structures. And it tells you about body position. So it tells you, for example, even without looking, if you put your arms out to the side, the receptors in your body are saying, hey, your arms are out at the side, because it's detected the movement and stretch in the joints and muscles that you used. They're called proprioceptors. And for most of these receptors, you don't have to be able to name the specific categories and types. For hearing, of course, we'll go over details, like I said, but for the other two, just recognizing that they're examples of mechanoreceptors. You have thermoreceptors, so the word thermo at the beginning should indicate to you that it's about temperature. The heat and cold receptors, most of which are on your skin, are really detecting variations of radiant energy, so they're detecting temperature. If you took chem, you might remember that temperature is the average kinetic energy of molecules in motion, and so things that are hotter have particles that move faster. That's why gases move faster and solids move slower. The last category that we'll take a look at are photoreceptors. Photo means light, so these are light receptors. The names of the photoreceptors are rods and cones in your eye, and we are going to look very specifically at those. But these are specifically detecting visible light. Whereas a thermoreceptor is detecting other types of energy, these ones are specific to the spectrum that we see as visible. So only particular wavelengths of light are going to affect our photoreceptors. Now those are the categories that we have. We're going to go into more details about photoreceptors in just a moment. So I just have a few slides here. We're going to start on the bottom left with the conjunctiva and sort of just go clockwise around the eye. So the parts of the eye are not listed in any sort of order of importance. They're just the order that the diagram has labeled them. So down here, this first one is the conjunctiva. And so that you can see, here's the little eyelash, and it's this bit of tissue just inside of it. And I'll just pause here now. Okay, so here's the conjunctiva down here. So it's not this piece that looks like an eyelash. It's the piece inside. Okay, that's the conjunctiva. It's the tissue that lines the inside of the eyelids, and it covers the sclera, or the white part of your eye. Now the next structure, because we're just going in the clockwise thing, is the suspensory ligament. The suspensory ligament is pointing to, if you can see this little red bit here, it's a ligament that is attaching what we'll see later on as the lens of your eye to the ciliary body. So this thing with the dots on it, it's the ciliary body. And this little bit of red, those are the ligaments that are attaching your lens to that structure. So the suspensory ligament is a series of fibers that connect the ciliary body of the eye with the lens, holding it in place. And we'll see in our next lesson that it also helps change the shape of the lens so that you can look at objects that are far away as well as objects that are close and still have them be focused. So we've got conjunctiva, suspensory ligament. The next thing on the list is the lens. So it's this big oval yellow and white shaped structure. My daughter was here with me on the weekend helping me set things up in the lab, and she loves looking at dissection stuff, so she had her eyeball out and was playing with it. And she was doing some tests on the lens of an eye to see how much force it took for her to crush it, because she likes to report these things back to my dad. So she phoned her grandpa and said, I couldn't do it, not with my hand, Grandpa. I had to use something hard. The lens is really firm. It's certainly not made out of the same stuff as the lenses of a pair of glasses, but it has the same function, and its firmness is not as hard as that, but still quite firm. It refracts light, which means it bends it to be focused on the retina, which we'll see in a few slides is at the back of the eye. So we have conjunctiva, suspensory ligament, lens, and then the next structure is iris. The iris is the colored part of your eye. So when you say that you have green eyes, blue eyes, brown eyes, we're talking about the iris of your eye. On the diagram, see this white thin piece that kind of extends over the surface of the lens? That's the iris. So it's not the part with the dots, but just in front of that, there's a skinny white piece kind of touching the lens, covers part of it. That's the iris. Now, aside from giving you beautifully colored eyes, it helps to regulate the amount of light that is entering your eye. So you actually saw this in action when we did our reflex activity, and you flashed a light in your friend's eye. The size of the pupil changed. But what really changed is what was going on with the iris, because the pupil is just the space in between the sections of your iris on your eye. So those are the first four, starting from the bottom, going upwards. I'll have the same picture on every slide, but we'll just talk about the next few structures. Does anyone have questions about any of the structures that you see on the list so far? Is it a question? No? So you can see, because we're going around clockwise, we're going to take a look at aqueous chamber, pupil, cornea, eyelash next. Yes. Sure we can. Thanks, Sarah. So picture's still there. Our next structure is the aqueous chamber. So this is the space. It's kind of pale yellow on this picture. It's in front of the lens, but behind this white segment here that's the cornea. So it's this little chamber, little compartment that you find at the front of your eye. It helps support the eye and the shape of the eye due to the pressure of the fluid that's inside of it. So the aqueous chamber is full of fluid. Now the next structure is this segment here called the pupil. The pupil is really an opening, so it's not a thing that I can touch. It's an opening. If you look at an eye from the front, because we're looking at it from the side, you would see the iris go all the way around the pupil. So the opening in the center of the iris, that's the pupil. And that's the opening through which all the light that you're going to see enters your eye. Conveniently, the pupil is located right directly in front of the lens, which we saw in the last slide was the thing that's going to refract and bend light so that you can see it. Now, in front of those two structures, this very outer layer, this pale colored layer in front, that's your cornea. Your cornea is the outer layer of the eye. It refracts light, not to the same degree that the lens does, but it still does. Your cornea is what most laser eye surgeries are fixing. My problem with vision prior to LASIK was that I was nearsighted, very, very nearsighted. And the reason I was nearsighted is because my eye's the wrong shape. They can't fix the shape of your eye, but they can change the shape of the structures that are refracting light. That's the whole principle of how glasses and contact lenses work. They adjust the way light is refracting to correct for whatever issue your eye itself has. So since they couldn't change the shape of my eye, they reshaped my cornea so that it refracted light correctly on the back of my retina based on the shape of my eye, which is why I can see without wearing glasses or contact lenses now. So that's your cornea, outer layer of the eye. Now, it didn't label it on the bottom, but I think you all know that you have eyelashes on the top and the bottom of your eye. So this one isn't labeled, but this one is, and I hope you can see they look the same. Eyelashes have as their job, aside from looking beautiful, but to protect your eyes from really small particles. And then I was reading this website. My husband likes this site called Dad Jokes, and he sends them to me all the time. And the one the other day was, how's this for irony? My eyelash is supposed to protect my eyes from things getting in my eyes. But whenever there's something in my eye, it's always a dang eyelash. And then I laughed, it was funny. Because eyelashes in your eye kind of hurt. Hey, they're irritating and they're hard to get out. Then the next structure, kind of like eyelashes, actually shows up on the top and bottom of the picture, but we're only labeling it on the top. It's the ciliary body. It's the thing with the little dots on it on the picture. You can see it up here, and you can see it down there. The ciliary body or muscle changes the shape of the lens when your eyes focus. So when you move your eyes from looking at your cell phone right in front of your face, which nobody's doing right now, to the smartboard up at the front, and back and forth because you're texting somebody or whatever, the ciliary body is constantly changing the shape of your lens, making it flatter or rounder, so that light gets bent properly in your eye so that far away or close up, things look focused. Now the ciliary body is attached to that suspensory ligament, which also shows up here. So it's all the way around the lens, but we're not going to label it every time that it shows up. So we've got aqueous chamber, pupil, cornea, eyelash, ciliary body. Anyone have questions they want to ask on any of those structures?
Right under pupil.
Cornea? Do you mean under pupil there or right here?
Right there.
So it says pupil, aqueous chamber, and then this line is pointing to iris. Yeah, you're welcome. Anyone else need to ask anything there? So if we keep going around, we were up here. You have an eyelid, and of course you have an eyelid on the upper and lower parts of your eye, but it's only labeled at the top. The eyelid is attached to your eyelash and is in front of the conjunctiva, because the conjunctiva was the inner lining of the eyelid. The eyelid is the skin that physically covers and protects your eye. So similar job as eyelashes, it's supposed to protect things from getting into your eye. The next structure is the sclera. Okay? The sclera is the outermost layer as we look at the round back part of your eye. So at the front, the outer layer is the cornea. As we as we follow the cornea towards the back of the eye, the part that you don't see from the outside that's in your occipital your eye cavity in your skull, follow the cornea and see that all that's happened on this picture is the color changed, but it's that same outer layer. So it's the cornea on the front of your eye, but as we follow that outer layer around the back of the eye, it's called the sclera. You'll recognize the sclera on the eyes we dissect on Thursday because not only is it the outermost layer, but it's a pretty firm, kind of beige colored layer, like it suggests here. It helps to protect and support the eyeball, and you'll see the firmness of that tissue is the main reason it does that. It helps maintain the shape. You have some muscles. There's one on the top there on the bottom. But you have muscles in your eye socket, the rectus muscle, for example, that help to move your eyes around. So not just to let light in and out of your eye, but to make it so you can look up and down and left and right and move your eye around in its socket. Quite frequently on the eyes we dissect, there's a little bit of muscle left on the outside, and it is sort of brownish, reddish colored, so you should be able to distinguish it. The next layer is the choroid. The sclera is the outermost layer. The next layer in, which is this dark colored layer here, is the choroid. It has a dark pigment in it to absorb excess light. You don't want light bouncing around on the inside of your eye. You want it to all be directed onto the photoreceptors that are at the back of the eye. It also is the part of the eye that contains blood vessels. And so if you think back to your knowledge of how cells work, they need a constant supply of oxygen, of glucose to function. So you need blood to transport things back and forth, and the eye is no different. Then we have the vitreous humor, which is this whole inner, round, pale yellow portion on the picture. It's the same as the aqueous humor, really, in terms of what it does. It helps support the shape of the eyeball, but the fluid here isn't as liquidy. The aqueous chamber, the consistency of it is really like the consistency of water. It's a liquid. But in the vitreous humor, it's more jelly-like. And so when you cut open the eyeball, like you will on Thursday, you'll see some liquid come out, but then there'll be a part where you actually have to scoop the jelly-like stuff out of the eye. So we have, where were we? Eyelid, sclera, rectus muscle, choroid, vitreous humor. We've just got one slide left with the other pieces, and I think we'll have to save that mystery for Thursday.